All right. So it's my pleasure to introduce our next speaker, Ariana Quinos. Works at Massachusetts Institute of Technology in biological oceanography. Thank you. Long day already. All right. Under Michael Follows. Good. And did a practicum at Lawrence Berkeley in 2021.
Great. All right, thank you so much. Today I'm so excited to talk to you about a totally different part of the Earth system, but still having to do with the global oceans, and that is single-celled microbial eukaryotes, or protists. So if you haven't heard of them before, protists are a key part of global biogeochemical cycles. They're both an essential link between the numerically abundant bacteria and higher trophic levels like fish and whales, and also a key part of global primary production. So protists include phytoplankton, photosynthesizing single-celled microbial eukaryotes that constitute over 50% of global primary production, all from the oceans. So they fill a really important role in understanding both how the ocean is operating ecologically, as well as playing this important part in biogeochemical cycling. But protists are really difficult to study. So they're microbial. We can't see them with the naked eye. We need some tools to be able to observe them to begin with. And in comparison to other other microbes, so other parts of this unseen world, they have very complicated genetics, and that's something that's quite relevant to my work in bioinformatics. So because they're eukaryotic, they have complicated repeat regions, non-coding regions of the genome, which can make them difficult to analyze bioinformatically. And over the past several years, we've started to have a growing appreciation of this concept known as the pangenome concept, wherein we know that even within one species, or what we might like to think of as a species, or what we can identify morphologically as a species, we have a lot of cryptic diversity in the form of this pangenome, whereby a species will share a set of genes, the core genome, that constitute them to be part of just one species, but then they all have these individual differentiated variable pangenomic sets of sequences that might be specific to one strain or another. And when I say strain throughout this talk, what I really mean is a cell that I picked out, propagated a lineage from. So I know it's part of this species, but it's really an asexually reproducing set of cells that all came from one original cell that I plucked out of the ocean sometime. And this genomic variability is just part of one axis of variability that we have to think about when we think of single-celled microbial eukaryotes in the ocean. So they're present in this dynamic oceanic context. So in addition to being taxonomically differentiated, both intraspecifically in the form of this pangenome concept and interspecifically, so thinking about these groups that you see up on the slide, coccolithophores that are calcifying versus diatoms that form glass-like shells. They're also present along different depth gradients. So most phytoplankton are in the sunlit layer of the ocean, the photic zone. But then deeper in the ocean, we have a lot of phytoplankton or a lot of protists that are heterotrophic, so they consume other organisms. So depending on what depth you find them in, they might have very different ecology. And they're also in a dynamic, moving ocean environment that's changing throughout time. So we have a time axis. There's variability occurring on daily scales as well as over seasonal and longer time scales. So in order to study these protists, we're going to need cross-scale tools and very different data sets. So the first are laboratory physiology experiments, whereby we can start to collect some baseline data about these lineages of protists that we're able to collect and study in the lab. We can also make field observations to help us understand these populations and communities. And finally, we can use ecosystem modeling. So we can try to place protists in a dynamic ocean environment and predict how they're going to behave. Our laboratory physiology experiments will inform our ecosystem models, as well as our field observations being used to both constrain and validate ecosystem models. And increasingly, laboratory physiology experiments and field observations can be linked by genetic sequencing. I can sequence what I have in the lab and use those results, those outcomes, to annotate field data so I can understand ocean ecosystems in a much more high throughput way than I ever have been able to before. And this generates a lot of data of many different types. So even in the lab, as opposed to in the past where I might have just collected some optical data to measure the growth of the cells in culture, now I can use flow cytometry every single day. The plate can be moved in an automated way so that I can measure hundreds of samples from laboratory experiments. And I can also collect data on optical properties of cells, so things like cell size, all from the lab on a daily scale, generating a fairly large amount of data compared to what I was previously able to measure. I have these large sequencing data sets, so both population level, so things like genomes and transcriptomes, either DNA content or expressed genes for these individual groups of cells, strains of different species, and also community level data, filtering large quantities of ocean water, sequencing everything that's present in that sample, and then trying to computationally resolve what organisms I had and what they were doing. And then finally, I have ecosystem model outputs, which both include the different members of the community as well as their ecosystem context, so the physical and chemical data that they find themselves in the ecosystem model. So the first thing I'll talk about are some laboratory experiments that I've been conducting in order to constrain some ecosystem modeling efforts. So this is a thermal block whereby I have a cool circulating water bath on the left and a heating element on the right, setting up a gradient of temperatures, and I can subject different strains to different temperatures over long periods of time to get a sense of the acclimated thermal response of different strains, and then link that to their genetic diversity using transcriptomics. And I subjected many different strains of this species of coccolithophore, Amelia huxleyi, to a range of temperatures in order to constrain their distributions in the model. So this is where those strains originally came from. They're found, or they were collected and made part of a culture collection. So I have strains that were isolated from all over the global ocean, from Antarctica to the equator, so very different thermal habitat. And from these experiments, I end up with these thermal response curves. So this tells me how temperature is driving differences in growth rate for different strains, and how that varies across strains that were isolated from very different original ecosystem contexts. And I can compare these results to theory that I have. So I expect, for example, that as thermal optimum's increasing, maximum growth rate will also increase. So in short, things that are grown at higher temperatures will tend to grow faster. But I have some major discrepancies from that general scaling here. A lot of my strains have much lower maximum growth rates than I might expect based on parameterizing that curve across all the different strains of that species. And I can use that in ecosystem model simulations where I can get higher resolution of the different thermal optima of different strains of coccolithophores and how those might change, to try to better constrain biogeography and identify discrepancies between what I expect and what I'm getting out of my model in terms of potential habitat of coccolithophores and how that habitat will change under future thermal conditions. Switching gears a little bit, I'm at the same time collecting these transcriptomes. So the circles up here are all the samples where I sequence transcriptomes, gene expression data for different strains at 3 to 5 temperatures for 7 strains. And these are really high dimensional data. So this is where some of the computing is going to start to come in. From just these samples for this transcriptome study, I have over 1.3 billion raw read sequences that were generated, and I've assembled those into over 981,000 transcripts on which there are over 1 million predicted genes. And this is just for one species and a handful of strains from a few locations. So there's a lot of diversity here. And so I'm trying to understand both what these different strains have in common as a species, as well as what differentiates them and makes them differentially able to deal with different thermal habitats in the ocean, as well as their potential to acclimate and evolve to future thermal change. And across all these strains, we see that they are mostly the same species. So the core genes are conserved the way we expect. They all align into the same region of the phylogenetic tree up there. But they have very different overlapping gene content. So while 24,000 genes over on the left here are shared across all 7 of the strains that I have, In just one of the strains, I have an additional 10,000 genes that one strain has and the rest of them don't have. And that happens to be the strain from the Southern Ocean near Antarctica. So it's clear that it has a lot of very unique genetic content that's enabling it to deal with these especially cold temperatures. So we're interested in exploring that gene content and also identifying it in the field later on. So switching gears to the field data that I have, which I'm using genetic sequencing in a totally different way in order to generate, a lot of tools are built for either bacteria or they're built for things that are not environmental, so not high complexity data the way that we have in the ocean environment. So in order to combat this problem, over the course of my PhD, I've built some tools which are specifically developed for this problem of eukaryotic microbes in the environment. The first is a metatranscriptome pipeline called Eurythmic, and the second is a conda-installable Python package for taxonomic annotation that's called Ukulele for eukaryotes, right? And so these couple of tools have been published over the past few years, and they're specifically for researchers that are working in these diverse environments. Users are able to automatically install databases, for example, for marine microbial eukaryotes specifically. And I applied this metatranscriptomic pipeline to some past environmental data sets or some previously collected environmental data sets, in this case from the Mediterranean Sea and the Southern Ocean. And what I found was that I was able to recover genes that not only were not previously recovered by other studies to be found in these particular samples, but were not previously recovered anywhere in the global ocean. So everything in blue here is part of that set of genes that were totally novel genes that were recovered by this pipeline and were not previously in any other ocean sample. And this is also a really big data problem. So just in that subset of samples on the previous slide, which was part of a global sequencing expedition, I had over 94 gigabytes of raw sequencing reads, required a lot of high-performance computing resources in order to assemble them together like a puzzle, predicted over 308,000 genes from just one of those assemblies and over 15 million genes from just that subset of samples that you saw on that last slide. So it's really a big data problem. Okay, and all of this leads us to the question of whether we're really accurately recapitulating these communities to begin with. I'm really in particular interested in this taxonomic annotation question. So in order to answer this question, I was lucky to have access to a data set where I had both microscopic observations for protists that we are able to observe and annotate to some extent taxonomically under the microscope, and I could couple that directly to some metatranscriptomic sequencing data. And what I found was that I had a big problem. So this group in the yellow here is a family of diatoms that was very abundant in the microscopy data, but I was not finding in the metatranscriptomes, even though I knew I had it present in my database. I had a laboratory transcriptome that should have been accurate for annotating this organism in the community level data. So this led me to the question of how I could develop better algorithms to be able to access this taxonomic information, to be able to accurately annotate the taxonomy of communities of microbial eukaryotes in situ. And this has led to some recent work using a two-stage pipeline to apply clustering algorithms, so unsupervised algorithms, to be able to get at the taxonomy more precisely, as well as to recover novel taxonomic annotations and to identify areas where I need to include additional sequences in the reference data sets. Because increasingly it's becoming cheap and easy to sequence additional samples to create new database entries, which is expanding the size of my database and making this an even more computationally intensive problem. Just in the database that I used for the pipeline where I was constraining and reanalyzing these data sets for this project that was shown on the previous slides, I had over 15 million reference sequences. And the way that I'm conducting this clustering is by using short subsequences from these assembled transcripts from these reference data sets. And depending on the size of subsequence that I use and how many different lengths I use, I might have up to hundreds of millions of subsequences to compare, as well as having to, if I'm just comparing the sequences one to one, I have to make on the order of 10 to the 14th computations. And these are computations that can be done in parallel. They're the type of computations that we really easily can leverage high-performance computing in order to speed up. So in short, we can use high-performance computing and high-dimensional data sets to make important ecosystem insights about, or to make important ecosystem findings about microbial eukaryotes in situ. So we can start to assemble maps like these, whereby we can take that whole massive ocean gradient, start to analyze it in a high-throughput way, and identify which major players are present in different regions of the oceans across depth gradients. And this can tell us a lot about the ecosystem services these organisms are providing and their biogeochemical functions. In particular, via sequencing data sets, at the same time as identifying taxonomy, we also can identify functional potential of individual microbial eukaryotes. So with that, I'd like to thank my advisors at MIT as well as at Woods Hole Oceanographic Institution, the many members of my lab that have helped with this, as well as my collaborators at the Joint Genome Institute at Lawrence Berkeley National Lab, who I continue to collaborate with, in particular on the taxonomic annotation dimension of this problem. I also like to give a shout out to the couple of undergraduates that have worked directly on aspects of this work, Wei-Xuan Li and Quinn Perrion, and of course to the CSGF for the opportunity to present here and for the help along the way. Thank you.